Hi, my name is Liz Brunk, and I'm an assistant professor at University of North Carolina, Chapel Hill in the Department of Pharmacology and Chemistry, and I received the PhRMA Foundation Faculty Starter Grant in Drug Discovery. I started to get into research during my undergraduate days, and I had a series of amazing mentors who encouraged me along the way. It wasn't until I got to the University of California, San Diego, that I started to really get interested in omics data. And omics data, it started with the human sequencing revolution, where we could sequence all of the genes in the human genome. And then we began to understand the RNA and the protein levels in different cells and systems. And I began to see how powerful it is that we can gather all of this data and start understanding how we can use this to interpret the impact of variants or the impact of drugs on these systems. There are many different types of variants across diseases. In particular with cancer, there are a lot of different types of variants we can see that are happening across patients. And the whole goal of precision medicine is to understand the impact of these variants so that we can design better therapies that match these patients' tumors. My lab is really interested in understanding the impact of variants, especially one type of variant called extrachromosomal DNA. When we learn about DNA in high school and in college, we think about all the chromosomes in our cells that hold all the DNA that is responsible for who we are and how our body works. What we don't learn about is that there are DNA fragments that are amplified outside of our chromosomes. And when cancer takes over, these DNA fragments can get out of control, and they amplify genes that are cancer-causing and promote fast growth of cancer cells. Cancer patients that have this extrachromosomal DNA in their cancer cells happen to be the hardest to treat cancers. They are the most malignant, and they have the most resistance to conventional therapy, and we currently don't understand how to target these. A part of my research is developing and using artificial intelligence and machine learning algorithms to help us to predict the variant impacts in these cells. We can use large-scale databases that have collected omics data across hundreds of thousands of patients and samples, and we can use the algorithm to learn what are the important features of this data that can predict how many of these patients have extrachromosomal DNA in their cancer cells, so that we can understand the prevalence of this as well as what we can do about it. How can we create new therapies to treat these patients? So receiving the PhRMA Foundation Award is truly instrumental to getting my lab's research off the ground and running. As a junior faculty, I'm an early career research scientist, and so much of getting future grants is having an idea and the evidence that we can do this research. We have to prove ourselves in order to get grants from NIH or other funding agencies. And it can be really hard to reach that point, which is why funding early career research scientists through these types of foundation awards is so crucial to helping science advance. I remember a turning point in my career was the first time I performed an experiment and was actually able to see extrachromosomal DNA in these cell nuclei using a fluorescent microscope. And this was a pivotal moment for me because I had up until that time been trained as a computational scientist who had only been doing research behind a computer. So it was all very new and scary and exciting to learn how to do experiments, work with cells, and then image them under the microscope. So when I sat there in that dark room looking under that microscope, it looked like a firework. There were all these different fragments that had been stained with different fluorescent dyes, and it was incredible. And even more incredible was the fact that it had been computational work together with omics data that had helped me to predict that extrachromosomal DNA was in this cell line. So we're really excited about the potential of AI in biology and medicine, especially as we are entering into this single-cell genomics era. So the future goals of this research are to create algorithms that can help us to understand what's happening in these single cells. We can begin to better design therapies that can match to patient tumors and be truly precise in the era of genome medicine.